Hello everyone, myself Roshni Praful Gandhi, and today we are discussing the very interesting topic, that is biotransformation and production of secondary metabolites. Then in this presentation we are discussing the variety of contents like introduction, advantages of biotransformation, ideal properties of plant cell, then examples of biotransformation reactions, and last is production of secondary metabolite, that is chiconin production. Starting with the definition of the biotransformation. Biotransformation means it is the process in which undesired product is converted into the desired product. That is, by using living cells, or these living cells are microorganisms, we can convert or change the functional group of organic compound and convert them into the chemically different product. Then it involves plant cell culture, hairy root culture, and immobilized cells. Then this biotransformation process, it involves variety of reactions as per the mechanism involved in the biotransformation reaction. They are named like reduction, oxidation, hydroxylation, acetylation, esterification, isomerization, glucosylation, methylation, demethylation, and last is epoxidation. Then moving towards the advantages of this biotransformation reaction. The first one is these biotransformation reactions are target selective, then specific, and they gives a chirally pure product, means they gives a pure product one. Then second one is the great potential to generate novel products, means this biotransformation reaction, as we heard, it deals with the synthesis of new product. So it obviously it provides a great potential to generate novel products. Then improvement in productivity of desired component. As definition indicate, it produces undesired component into desired one, so it improves the productivity. Some next advantage is sometime it is cheaper to use microorganism to prepare an organic compound than to synthesize it chemically. See, if we synthesize any organic compound, various chemical processes are involved, or various stepwise processes are involved, and these are various tedious process or complicated one, and it involves increases the cost of the product. So as compared to chemical synthesis, as we follow biotransformation reaction using microorganism, the same reaction is carried out using faster or cheaper rate. Then last advantage of this biotransformation reaction is production of secondary metabolites and various pharmaceuticals. We see this in detail. Then ideal properties of cell culture. What ideal properties the cell culture bears to withstand or to carry out the biotransformation reaction? First property of the culture medium is it must contain the essential enzymes which are required for reaction, because enzymes play the role of catalyst or enhances the rate of reaction. Second, the substrate or precursor should not be toxic to cell culture. Obviously, as the substrate is or initial component or raw material is toxic one, so the final product is also be a toxic, so the substrate must be non-toxic one. Then next, third one, that is substrate should easily reach reaction without degradation. The substrate should not decompose in the reaction mixture. As it decomposes, it means it loses the productivity. So the substrate should not decomposes in the reactions. Then next, rate of product formation must be faster than its metabolism. The rate of synthesis of product formation is faster one. And last is the culture must tolerate the added precursor and the products. The culture medium should withstand all the contents or raw material which are added. Next, examples of various biotransformation. See the first one. In this, we use Paganum hamela cell culture. The cell culture is Paganum hamela cell culture. And this cell culture medium, what it converts? It converts geranyl acetate to geraniol, and another is linyl acetate to linanol. And the plant species used in this process is Rosa centifolia. Okay, by using that mediums or this reaction occurs. Then next one is biotransformation of certain cardiac glycoside, that is cardiac glycoside digitoxin, it converted into digoxin by using the plant species, that is Digitalis lanata. Here the culture media name is not mentioned, but you remember that is this digitoxin, it is converted into digoxin by using the plant species. Another is the biotransformation of some steroids. Steroids are also converted by using, for example, cholesterol, it is converted into diosgenin by using the plant species, that is Dioscorea bulbiferia. So this diosgenin and digoxin, these are the important one as per pharmaceutical industry. Another steroidal conversion, that is progesterone, it is converted into 5-alpha pregenin 320 dione. In this, progesterone is converted by using living cells, that is Digitalis purpurea, and the mechanism or type of the biotransformation reaction is reduction reaction. Here some other detail of the biotransformation examples are given mentioned in table, that we see. For example, testosterone converted into 6-beta OS testosterone. Then quercetin, it is converted into quercetin thioglycoside. Then salicylic acid, it is converted into isosalicylene. Coumarin, it is converted into umbelliferone. Okay, and the reaction involved, that is further like hydroxylation, glucosylation and hydroxylation. And the different plant species are used. For example, in the testosterone conversion, M polymorphia species is used. Then quercetin, cannabis sativa is used. Salicylic acid, dhatura species is used. And coumarin, that is Catharanthus roseus plant species is used and converted into umbelliferone. And these are the categories of the substrate. For example, testosterone belongs to steroidal category. Quercetin, it is a flavonoid. Salicylic acid, it is a phenolic acid. And coumarin, it is a plant glycoside. Then next, production of these secondary metabolites. Okay, plant produces two types of metabolite, that is primary metabolite and another is secondary metabolites. This primary metabolite, what its roles? See, plant produces primary metabolites which are essential for normal growth and survival of plant species. Means these primary metabolites are essential for the normal functioning of plant, that is normal growth and maintenance of the plant. But what is the role of secondary metabolite? Next, the secondary metabolites produced from the plants are not involved in plant normal growth but play significant role in plant defense mechanism. What are their functions? They are not involved in the normal growth of the plant, but they play important role in plant defense mechanism. But these secondary metabolites, these have variety of pharmaceutical uses, like it has therapeutic antimicrobial activities. So as per pharmaceutical industry, these secondary metabolites are important for us, we peoples. But one question is here, that is this one problem: these secondary metabolites which produced by plant are activated only at the particular stage of plant growth, and it is essential to prepare secondary metabolite using biotransformation technique because of its pharmaceutical use. Okay, and the problem is that these secondary metabolites are useful for we peoples, but the production of this secondary metabolite, it is limited one. So by using this biotransformation technique, we produce this secondary metabolite by synthesis method, and ample amount of secondary metabolites are produced one in industrial purpose. Okay, then our last content, that is production of chiconin or secondary metabolite chiconin. It is a red naphthoquinone pigment. Red naphthoquinone pigment means it involves red color and naphthoquinone category. Then the secondary metabolite chiconin has two types of activities or actions, like antimicrobial and anti-inflammatory action. Another use of this secondary chiconin is it also promote wound healing, then skin ulcer and hemorrhage. Another use is in cosmetic industry. This chiconin is useful for production of lipstick, lotions and soap. So these are the three main roles of chiconins for production. Then for production of chiconin, we use the plant species, that is it is prepared from the roots of Lithospermum erythrorhizon, a perennial herb. This herb is located at Japan, Korea and China. Okay, the plant species used for the production of chiconin is Lithospermum erythrorhizon, and the media, that is culture media used for production of chiconin is Linsmeyer, Scooch, Basel, agar media. In short, it is called as a LS media. Remember, that is LS media is used, and another is Lithospermum erythrorhizon species. Then these are the factors which influence the chiconin biosynthesis, or these are the precursor factors which are added or maintained at the culture media. First factor is temperature. We must maintain the temperature of culture medium in between 20 to 25 degrees Celsius, and dark condition is maintained so that to increase the cell growth and yield of the product. Okay, then second is growth hormones. These are also essential part. The natural auxins like indole acetic acid, IAA and kinetin, they are added to culture medium to increases the growth of the chiconin. Another is carbon source of carbon. Obviously sugar material is added as a source of carbon, that is 5% sucrose. It is best source for maintaining the growth of the tissue. Next, source of nitrogen. The nitrogen source added in the media is 20 molar NH4 and 40 mm NO3. As a nitrogen source, we are included it in culture media. The next content of the culture media is rosmarinic acid. The function of this rosmarinic acid is the extra cells which do not produce chiconin. These cells are accumulated by this rosmarinic acid, and we can further remove it. Another is source of polysaccharides. Agar powder is added to media to induce culture. Okay, then ammonium tend to inhibit chiconin synthesis in absence of agar. Okay, agar is as a polysaccharide source, we add agar powder. And last component are various concentrations of Cu, that is copper, NO3, Ca, calcium, iron, boron, SO4. These factors increases the yield of chiconin, or these are the parameters which are added to culture media. Next, moving towards the production of process of chiconin, two-stage culture system is developed for high yield of product. The culture system of chiconin involves two stages. At first stage, we use Mg5 media. We are seeing this in detail in next slide. That is, here it remember that first stage we use Mg5 media, and another at second stage we use M9 media for chiconin production. Next, chiconin yield increases when initial cell density and medium concentration were doubled. It yields increases when we increases the concentration or of medium. Then the another important parameter in this process is we maintain the oxygen level of the product media at both the stages, first and second stage. We adjust the oxygen up to 6.4 to 6 ppm so that suitable growth must be done. Another is we use the two medias, that is Mg5 media. It has 200 liter capacity, and here it we incubate it for 9 days, and the production of M9 media is powered, that is M9 media, it is of 750 liter capacity, and we culture it for 14 days. Okay, then after seeing the next slide, you are totally gaining the topic. That is, after incubating cells in second tank for 14 days. Sorry. Okay, after incubating cells for 14 days in second tanks, this chiconin is harvested or collected by using filtration technique. After collecting it, after filtration, the extraction and purification of the chiconin is done so that it converted into pure form. With the extraction is done with anhexane and subsequent hydrolysis with 2% KOH and recrystallize to give pure chiconin product. See in the image, you are seeing that the red color pigment, this is the chiconin. Okay, then see we see the detail regarding large scale production of chiconin. Here we are using two medias. As first I tell you, that is Mg5 media and Mg9 media. In the first Mg5 media, it is, or see the first tank and second tank. First tank it is of 2 liter capacity, and second tank it is of 750 liter capacity. Here this Mg5 media is incorporated into the first tank, and the cells, that is lithospermum erythrorhizon cells, are placed into the first tank. Media and cells are in contact with each other, and here oxygen level and other parameters are maintained. Okay, then incubation is done. Then this first Mg5 medium content is filtered, and then here Mg9 media, that is fresh media, is faced with the cells. This stage is our first tank process, takes 9 days for preparation of the chiconin. After 9 days of chiconin production, this chiconin is transferred, or chiconin cells are transferred into the second tank for further growth of cells. That is, in the second tank, here it is placed up to 14 days. and after the completion of time period, that is 14 days, these cells, white cells, are converted into red pigment, or they gives a red pigment, that is chiconin or red naphthoquinone pigment. This red naphthoquinone pigment, it is still unpure form, so it is filtered by using filter medium and processed by using an hexane or KOS solution, and we getting the final product, that is chiconil. And last one is, thank you for listening me carefully.